Okay. My name is Valentina Losardo. I'm an assistant professor in the same department as Rupa, actually, cell and genetic biology. Our lab studies mostly genetic risk factor for cardiovascular disease. And we say our lab is a functional genomics lab, and we use mostly stem cells to study cardiovascular risk. So today, Elsa is presenting her work. She just finished her third year. Elsa has one big accomplishment to be my
Only one.
first student. The first is she's my first student. I'm super proud, but she's been also amazing to keep up with a young investigator, I would say. She's doing fantastic. Her work, I'm hoping you will appreciate the amount of work that she has done in only 3 years. Her paper is currently in bioRxiv and just under review in one of the American Heart Association journals. So I'm very proud. Yeah. And Elsa is a great lab member. I think that she's extremely dedicated. She's very patient, which I think helps a lot. And she's extremely creative, which I think is very refreshing in graduate student. And then, without further ado, Elsa, take it away.
Hi, everyone. We'll get going because I am all that stands between you all and the picnic. I know my place here. So we study coronary artery disease. This is a disease that is characterized by the formation of atherosclerotic plaques in the human coronary arteries. And it is traditionally thought of in connection with lifestyle factors like diet or exercise, but it actually has a very strong genetic component as well. It's about 40 to 60% heritable, and 300 genomic loci have been linked to risk for coronary artery disease or CAD. Of these, a full quarter are predicted to affect the vascular wall, as opposed to just 10% being lipid related, which I think would be counterintuitive for a lot of people. And then around 50% are unknown functions. So of these loci, the very first to be linked to coronary artery disease was 9p21.3, and it was identified in 2007 in a series of GWAS studies. And it remains to this day the strongest single genetic factor, risk factor for coronary artery disease. And it's a pretty interesting region for them to have found to be so impactful because it is not only a non-coding region, it's actually a gene desert. So it doesn't contain any genes at all. It's about 60 kilobases long, and what it does have are single nucleotide polymorphisms, or SNPs. It has about 80, and they are in very, very strong linkage disequilibrium. And this linkage disequilibrium creates a haplotype with 2 alleles. So one allele confers this increased risk of coronary artery disease, about 30% increase for every copy that you have, and then the other does not confer any increased risk. It is worth noting that although this region does not contain any genes, it does overlap with this long non-coding RNA ANRL. Now, although this region was identified almost 20 years ago now, there hasn't been as much research as you might expect into it. And part of the reason for that is because of the limitations with animal models. So this is a region that's only conserved in higher order primates and humans. So we get around this, not using animal models, by using human cells. So we take peripheral blood mononuclear cells, somatically reprogram them to iPSCs, and once we have those stem cells, we can make any type of cell that we want. This also has a notable advantage of maintaining the genomic background of interest that we want, while creating cultures that we can maintain almost indefinitely, as opposed to primary cells, which will senesce eventually. So this allows us to genetically edit those cells and do functional studies. Now, as I mentioned, we can make any type of cell that we want from our stem cells. And in our case, the cell that we want are vascular smooth muscle cells, or VSMCs. These are a type of cell that exists in the human arterial wall, and it plays multiple central roles in the progression of coronary artery disease. They're known to invade and accumulate in the intima, the innermost layer of the artery, in the early stages of the disease. And by the end of it, the average atherosclerotic plaque has about 70% of the cells in that plaque coming from vascular smooth muscle cells. As they accumulate and they form this plaque, they also go just hog wild. They do all kinds of stuff that they're not supposed to be doing, including transdifferentiation into fully other types of cells, such as macrophages or foam cells, and also modulation of their phenotype, where they kind of take on traits of other cell types, but they don't fully transdifferentiate into those cell types. There's been a variety of these kind of modulated states reported in connection with coronary artery disease, many of them initially reported in mice and now more recently in humans, for example, fibromyocytes, SEM, and fibrochondrocytes. So what we do in our lab is we take donors who are homozygous risk or non-risk, one of each, and we get their peripheral blood mononuclear cells, make our iPSCs, and then we genetically edit those iPSCs to completely knock out the 9p21.3 risk region. So 60 kilobase is gone. And that way we have isogenic knockouts in both backgrounds, creating these 4 genotypes, which are going to keep coming up a lot throughout this presentation. So good to bear in mind: the risk intact cells, the risk knockout cells, and the non-risk knockout cells. And once we have those genetically edited iPSCs, we can then differentiate them to vascular smooth muscle cells and start kind of digging into the differences between the genotypes. So we published with this system in 2018, and one of the first things that we looked for were smooth muscle cell markers, just to make sure that everything had differentiated correctly from iPSCs, especially our knockout cells, which are missing quite a large portion of the genome. And as you can see from this staining here, all the genotypes look pretty similar, so it would appear that everything differentiated correctly. But when we dug into the transcriptome with bulk RNA sequencing analysis, we saw some huge differences between the risk cells and our other genotypes. So in this panel that just popped up, this red group here, those are the risk intact cells. The 2 green ones are our non-risk background lines, and then the yellow is the risk knockout. And you can immediately see the risk is off by itself. Very interestingly, the risk knockout, the yellow group, is much closer to our green groups, the non-risk groups, than it is to the risk groups, indicating that the deletion of the locus was enough to almost completely revert those risk cells back to like the non-risk phenotype. This transcriptomic difference that we're seeing here between the risk cells and everything else is a difference of more than 3,000 genes, so quite a large one. And when we dug into it, we found a bunch of genes related to muscle contraction and biological adhesion. Both are functions you would expect to see in happy, healthy VSMCs. And when we dug into the functional assays of these cells, we saw corresponding defects with reduced adhesion and reduced contractility in our risk cells. And then over here in these images, you can see that the risk cells are also morphologically different from the non-risk and other genotypes. They're much smaller. So coming out of this work, we concluded that the risk region induces this 3,000 plus gene transcriptomic change, which includes many adhesion and contraction genes. It also induces corresponding functional defects like reduced adhesion and contraction and morphological defects. And finally, that the deletion of this region essentially reverted the transcriptomic profile almost all the way back to the non-risk. So my goal with my project work going into this work is to see if there's any connection between 9p21.3 and any of those like weird coronary artery disease cell states that have been reported in vascular smooth muscle cells, and to see if I can't start describing a mechanism by which 9p21.3 acts to create this impact. So what I did was I took the cells of those same 4 genotypes, the risk intact, the risk knockout, the non-risk intact, and the non-risk knockout, differentiated to VSMCs, and this time we sent them for single cell sequencing. Now you can see my data up here in this UMAP. Thank you, Harshi, for introducing what a UMAP is. These dots are all cells. They are colored according to clustering. Cells in the same cluster should be expressing similar genes in a similar way. So you can see we ended up with 9 clusters here. And once again, one of the first things we did was look at smooth muscle cell markers to see how they were expressing in our data. But now we were able to compare with a published single cell data set taken from endogenous human arteries. So these are ex vivo cells over here. And you can see that this group, these are the smooth muscle cells. And when we compare the expression of these markers in the endogenous smooth muscle cells to how they express in our iPSC-derived smooth muscle cells, you can see that it's really very similar. So this not only shows that our cells correctly differentiated to VSMCs, it also shows that these cultured VSMCs are reflective of cells that you might find in an actual human artery, and therefore something that's useful for modeling disease, and not just like a series of weird stuff that happens when you put cells in a dish. To further support this, we collaborated with Clint Miller's lab over at UVA, and they have this wonderful atlas built from multiple single cell sequencing data sets from multiple publications, both healthy arteries and atherosclerotic arteries. And we asked them to project our iPSC-derived VSMCs in this atlas to see where the overlap was. So the atlas SMCs are there in the middle in pink, and then the iPSC-derived VSMCs from our lab, they're here in gray. And you can see there's very strong overlap, once again, supporting the validity of our model as something to model disease. So the next thing we wanted to see was where our genotypes were grouping in this data. So here I have, once again, our UMAP of the data colored by cluster, and then over here on the right, I have it colored by genotype. And you can see that our two non-risk background lines and our risk knockout line, or cells, they're all kind of intermixing in these three portions of the UMAP. And when we look at where the risk cells are, we see that the vast majority, this is about 75% of the cells, are off by themselves in this portion of the UMAP with very little overlap. And our next step was to see how this pattern played out with respect to our clusters. So what clusters are hanging out in this portion of the UMAP? And what we found was that clusters 3, 4, and 6 are the ones that are kind of our risk clusters. And that's kind of what we moved forward with in our continuing analysis. So then I did some gene ontology to see what biological functions were represented in the lists of genes that were driving each of these risk clusters. So I have clusters 3, 4, and 6. And then I have on the left our upregulated genes, and on the right are downregulated genes. And immediately we noticed a pretty interesting pattern. Over here in the downregulated genes, there are several terms that you would expect to see upregulated in happy, healthy VSMCs. But here they appear to be repressed, including wound healing and cell adhesion. And then over in the upregulated terms, there are several things that you would not expect to see in happy, healthy VSMC, such as cartilage development, and then cluster 6, which is just straight neuronal development. Unclear why. But we did take away from this that clearly our vascular smooth muscle cell, the risk cells, despite expressing SMC markers, are taking off in a direction that is not like the other genotypes and definitely not behaving normally. So to confirm this even further, we did a trajectory analysis. So we take our single cell data, we feed it to a program that assumes that there is a transition of some kind in this data, and we ask it to identify where that transition is. So the starting end of the transition is going to be in dark purple and the end is going to be in yellow. And we did this separately in each genotype. So you can see we anchored the beginning of the trajectory in cluster 2, which has approximately equivalent representation of all the genotypes right around in this area. And as the trajectories proceed from left to right, you can see that in the non-risk and in the two knockout lines, they tend to kind of go down as they proceed towards the right, with the non-risk ending up here in cluster 0, and the two knockout lines ending up right next to it over here in cluster 7. Whereas when you look at the risk cells, they kind of go upwards as they go towards the right of the map, ending up right around here close to clusters 6 and 5. So once again, confirming that there's some kind of divergence between the risk cells and our other genotypes. We also looked at the genes driving these trajectories and found that these divergent trajectories are driven by kind of different modules of transcription factors. So these red ones are only present in the trajectory for the risk. The green ones are common to the other 3 genotypes, and then I had 2 orange ones that were unique to the knockout lines. So seeing that these cells were expressing SMC markers, but also clearly not behaving normally, we wanted to compare back to these kind of previously reported modulated VSMC states in connection with CAD to see if 9p21 was creating gene or a phenotype that resembled any of those. So we looked for transdifferentiation to macrophages, and you can see here I'm comparing these macrophage markers and how they express in an endogenous macrophage population from this healthy human artery data set. Those are the 2 macrophage clusters lighting up nice and bright with those markers. And then those same markers are not present at all in our data. And we did this with many more markers and many more cell types and found no evidence of transdifferentiation. So we didn't think we were heading in that direction. We also checked for some transitional disease states. So these are disease states where they don't fully transdifferentiate into another cell type, but they do take on features of other cell types. In this case, we looked at fibromyocytes, and this is an atherosclerotic human artery dataset, also from an endogenous human artery. And they have their population of fibromyocytes right there in that little green cluster. And once again, the marker for that cell type is showing up nice in their data and not at all in ours. So it doesn't appear that we are seeing fibromyocytes in our cells either. We also checked these classic VSMC modulators, MyoCD, SRF, and KLF4, to see if they could explain the differences between our genotypes. And as you can see here, this the violins are quantifications of the patterns that you see in the UMAP. They're not localizing really to any cluster or genotype, so it doesn't appear that they are explaining the differences here. However, we did a transcription factor analysis, and we saw enrichment for this transcription factor, SOX9, which is known to play a role in the mediation of osteoblast and chondrocyte genesis, as well as having been reported in connection with a disease version of VSMCs in which they take on features of osteoblasts or chondrocytes. So when we saw this enrichment, we wanted to check for other indicators of this modulated state. And sure enough, we saw concomitant enrichment for collagen 2 and a decrease in collagen 1, also indicators of this kind of osteochondro phenotype. Finally, we checked for actual markers of osteoblasts and chondrocytes, and we did see an enrichment for them in our risk cells. So from this, we concluded that it seems like our cells are starting to approach something like an osteochondral-like state. Previously reported modulated states that take on features like this are speculated to be related to arterial stiffening or calcification that we see in late-stage coronary artery disease. So potentially something to do with that. To continue to support this, we returned to the data from our collaborators over at UVA in the Miller lab. So they did an analysis where they took their smooth muscle cells from their atlas. So this is just their data, not ours is not in here. And they did a trajectory analysis, and they found that this trajectory was headed towards this population of cells, fibrochondrocytes, which are only present in lesion arteries, not in healthy arteries. And these are cells that you might guess from the name have features of chondrocytes. So seeing also suggestions of something like osteochondrocyte-like features in our cells, we wanted to see where our risk cells were placing in this trajectory analysis and whether there was any overlap with this previously reported modulated state. So this is the integration of our iPSC-derived VSMCs with their smooth muscle cells, and then we included fibroblasts for context. And you can see the iPSCs are in gray, they do overlap with the SMCs. And here it is again, this time the atlas cells are color-coded by the subpopulations found within the different cell types. And you can see that there's quite a lot of overlap between our cells and this pink SMC population, and those are what they call transitional SMCs. And then they did the trajectory analysis with our cells included. Now you can see the trajectory proceeds from the top to the bottom of the UMAP, and the fibrochondrocytes are here towards the bottom of the atlas cell group. Most of our cells overlap with the transitional SMC population here, and then we have this kind of arm that extends below the fibrochondrocytes. And if you look at the breakdown of that portion, it does include some cells of all our genotypes, but it's really mostly risk cells. And these cells are proceeding along a parallel trajectory to the fibrochondrocytes. They're also heading towards the bottom, but they appear to go much further along this trajectory than the fibrochondrocytes that are found in the endogenous human arteries. So we speculate from this that our cells are taking on an even more severe version of the fibrochondrocyte-like state that is seen in this data set. Now, why this more severe phenotype doesn't show up in the native arteries could be a couple things. One, it could be technical difficulties if there's some kind of unintentional bias in how they're extracting the cells from the tissue that doesn't allow for them to catch this kind of more severe phenotype. There could be something in our culture conditions that is prompting the cells to proceed further along this trajectory than the native cells do. Or there could be something in the microenvironment of the endogenous cells that is proverbially putting the brakes on them up here before they can get down here. Whatever the case, to us, this suggested pretty strongly that our cells are showing strong indicators of resembling this fibrochondrocyte-like phenotype. So I mentioned earlier that there are actually 300 loci in the human genome connected to coronary artery disease. So having seen that we're taking on indicators of this previously reported state, we also wanted to see if it might interact with any of these other loci that have been reported in connection with CAD. This is a lovely Manhattan plot from a 2022 publication of all those loci. And here are violin plots showing a bunch of genes at these locations that are differentially expressed in the presence of the risk haplotype. And you can see there's quite a few of them. So this suggested to us that the risk phenotype may be a cumulative product of many effectors rather than kind of a simple on-off switch that's easy to kind of boil down to one. So we wanted to look for other unique features that may be driving the state that haven't necessarily been reported in connection with coronary artery disease. Since we have this lovely single cell data set with isogenic knockouts, and we have the ability with this model to draw a really clear cause-effect connection between the 9p21 locus and any new genes. So we looked at the top genes that are differentially expressed between the risk genotypes and the other genotypes. And we saw these two genes, LIMCH1 and CRAB-P1. And these top two figures, the UMAPs and the violin plots, these are how they express in our data. the single cell data that I've been talking about. But down here, these are qPCR bar charts to confirm this enrichment in additional donors, because our single cell data is based off of just 2 donors. So here we have 2 additional risk and 2 additional non-risk donors. And these were actually done by Carol, who is right there. Hi, Carol. Thanks for doing those qPCRs. We also confirmed this with staining, comparing the risk cells and the non-risk cells, and you can definitely see LIMCH1 and CRAMP1 are super enriched in the risk. And this was done by former lab mate Rohan Zadi. And finally, I wanted to see kind of where the overlap was between these 2 genes, LIMCH1 and CRAMP1, and then SOX9. So here in this bar chart, all of these little purple regions, they're all LIMCH1 positive. And then the different shades of purple are kind of how LIMCH1 is occurring with SOX9 and CRAMP1 in different combinations. And so what we're seeing here is, it looks like LIMCH1 is kind of the big definer of the risk state together with SOX9 and CRAMP1. They kind of seem to be what's really driving this. So from this, we concluded that iPSC VSMCs are reflective of the overall transcriptional profile of ex vivo SMCs, and we can use our cells to model actual disease. Our risk cells show a transcriptional signature that has minimal overlap with the other genotypes. It's unique. The isogenic risk knockout exhibit a signature similar to non-risk cells, demonstrating that the deletion of the haplotype is enough to almost completely revert them to the non-risk. And we described for the first time a connection between this 9p21.3 risk allele and the osteochondromyocyte-like CAD phenotype previously reported in VSMCs. And we demonstrate this to be a cell autonomous process. We also show that the risk VSMCs appear to comprise an advanced stage of this osteochondromyocyte-like phenotype in the pseudotime trajectory. And we uncover 2 novel signature genes for the risk genotype, LIMCH1 and CRAMP1, which combined with SOX9 expression appear to define the 9p21.3 risk status. So that brings me to the end of the science that I'll be discussing with you all today, and to my RCR topic that I have chosen, which is scientists as responsible members of society and the social impacts of scientific research. And this is something that I chose because I'm super passionate about it, but also because as genetic technology becomes more and more a part of everyday life, the responsibility to kind of be thinking about the impact of how we present our research is going to be more and more pressing. This is something we think about particularly in the Sardo Lab because of the nature of the region that we study. So I mentioned earlier that this region is in a very strong linkage disequilibrium, and this linkage disequilibrium defines the risk and the non-risk versions of this region. What I didn't mention is that this linkage disequilibrium is present in all the world's populations except populations of African ancestry, where the linkage disequilibrium is almost completely lost, as you can see in this linkage disequilibrium map. Now, this is clearly an equity issue because people who have this ancestry aren't going to be able to benefit from research into the 9p21.3 locus the way that everyone else is. And this is not something that's unique to 9p21.3. This is something that crops up in a lot of loci that are linked to coronary artery disease and cardiovascular disease in general. So it's definitely something we want to be talking about because this is something that we want to be addressing. But it's also tricky to talk about because race and ethnicity are socially constructed concepts with no basis in biology and have been historically used to perpetuate some really pretty harmful narratives in terms of societal impact. Fortunately, we live in times where the need to kind of think about this and talk about this is pretty widely recognized. And so we have tools to help guide scientists who are trying to present research in terms of how they should be discussing that research. For example, this chart from the National Academy of Sciences, Engineering, and Medicine's guide using population descriptors in genetics and genomics research. Can you see the text? Yeah? Yeah, it's close enough. So up here on the left, we have the different types of genetic studies. Up here on the top, we have the different kinds of terminology. And you can see race terminology almost never recommended. Where the Sardo Lab would fall, I think, would be under number 3, gene discovery, complex traits. And so we really try to stick with genetic ancestry when it's relevant and genetic similarity as we're discussing our results. And of course, as the science progresses and as representation in the field improves, this may be refined or even made obsolete. And I look forward to keeping up with those changes because I think it's really encouraging to see people kind of thinking about it and working to affect change in these areas where we have equity disparities. Okay, acknowledgements. Thank you to my PI and to my lab. They've taught me stuff. Thank you to my University of Wisconsin collaborators, Huan Yang and Junha Shin. Special thanks to two graduate students, Catherine Huber and Kagani and Yolazada, who helped me with the computational aspect. And thank you to my committee members for their support, my outside collaborators over at the Miller Lab, and finally to Simon Gurris for funding me.
That's a great talk. It's a ton of work. I have so many questions I don't even know where to start. I guess I'll just ask, what's the allele frequency of the risk allele?
The risk, it occur at least, I started like 4 sentences in 3 seconds. Around 50% of the world's non-African population has at least one copy of the risk allele.
Okay, so I guess this is a trait that affects a disease that happens long after reproduction, right? So it's not maybe
Yes.
accessible to natural selection.
Yes.
So are there any other GWAS traits, maybe more developmental, for which this allele has a signal, like where you might suspect that the risk allele has some sort of phenotype with another trait?
Another trait?
Yeah. Like, why does this risk allele? It's one of the rare alleles that has a huge effect size, but is very common. So perhaps there is some signature of selection for it. What might that be?
Well, we have speculated that it could promote a phenotype that can be protective in a sense in coronary artery disease. Well, not protective. So we see reduced migration, which I did not bring up at all in this talk. And it's been hypothesized that migration is kind of part of how the atherosclerotic plaque forms. But at the same time, it's also part of how the fibrous cap forms, which is protective. It's not really clear. As for how it affects other traits, that I'm not really sure. I know that this region has also been linked to glioma and diabetes, right? But as far as explaining how it became so common, like where it might be beneficial in a way that natural selection could act upon, that I'm not sure.
I think it's actually developmental other than height.
Height?
You say every?
That is not linked to any developmental trait, so that makes think that it's actually, a positive selection during development. The only other trait that is not a disease trait is maybe height. But I'm not sure how much that would, be important. But.
And then my second question is like, what's going on here? So you said that there's a gene desert, but maybe there's a non-coding RNA. Like, what are the kind of maybe molecular mechanisms?
Yeah, so that long non-coding RNA anrole, we do know it plays some kind of role in the risk phenotype. In our previous publication and another project that's going on in the lab, we do see a kind of partial induction of the risk phenotype when we overexpress certain isoforms of anrole. As for like beyond that, how are things happening? We're still kind of working on that because we know anrole only accounts for part of it. It doesn't completely reproduce the risk phenotype, but it does seem to be playing a role. It is also a region that's predicted to be enhancer rich, but we haven't really looked in that direction yet. It's on our list of things we want to do.